Okay, so we already talk about all these different pathways that are involved in the development of pancreatic cancers, and they're not only in pancreatic cancer but in other type of cancers. We're going to finish with cell adhesion. That is also very important. So I'm going to give a little review about cell junction, cell adhesion, and then I will give you talk about some process involved in when what happened when you reduce cell adhesion. So let's start. So first thing that I wanted you to remember is that to understand cell adhesion and junction, you need to remember what is an epithelium. Yeah, and epithelials are very flexible in terms of variety of shapes. So you can have tubes, yeah, that are whole. You can have structures that are flat. You can have other ones that bend. There is so many shapes, and during development, there is a lot of variation. Yeah, and generally, epithelials have an apical region. Yeah, a bisolateral region that is the here, and the basement membrane that we talked in a previous class. And here you have again the basal lamin, and here you will have the extracellular matrix. And if you remember, the integrins are going to be, I'm going to put them in blue here. They will be located here. Yeah, in each one of these cells, you will find the integrins here. But in addition of the integrins, I told you that there were other type of junctions. That this is these ones that you can see here. That we will talk about that, and they are also multiprotein complex, so a lot of protein. So in addition to the epithelia, yeah, to this one, you have also connective tissue in which the cells are embedded in the extracellular membrane. There will be also integrins here that they will be interacting to each other with the extracellular membrane. But here the key is that they don't have this apical and basal polarity. So what happened during cancer is that in many cases these cells turn into something that look like this, and they start traveling. So they have a complete transformation. And remember, this cell will have the same gene that these ones, so they just need to activate different type of genes. Okay, so remember, let me delete this. Epithelial, this structure that I show you, they are a group of cells that are polarized because they have this apical and basolateral region. Yeah, and here you have the apical. Let me delete all the things that I draw here so it's clear. So they have this apical and basal polarity. Here you find the basal polarity. The connective tissue it doesn't have this apical and basal polarity, and they are more loosely organized. Well, this one they form like bricks in a house. You know, one attached to the other properly, and here the cement will be these proteins that are the junctions. And um, so in the connective tissue, they will be attached to a rigid scaffold, into a in rigid scaffold. And here you can see in more detail these cell junctions. So again, apical, basal, and basal membrane. Here they could be the connective tissue, the basal membrane, and here all these things are junctions that again are like the cement that allow the cells to attach to other ones. Other ones work like windows that they allow to communicate, or doors that allow to communicate between different cells. And in cell biology, we will talk about that in detail. It's like an hour, 2 hours of class talking about cell junction. So I'm not going to go into a lot of detail. I'm just going to tell you that multicellular organisms, they join cells, and how they do that? They do it by using junctions, yeah? And they have this specialized structure that are multi-protein complex. And here you have adherent junctions, desmosomes, tight junction, and gap junction. And there is no need that we talk that you memorize everything. Yeah, that these ones that all of them are important, and this one are involved in the adhesion. Okay, and all of them have some similarities. All of them are junctional complex, so they have many proteins involved. That so here I will say multi-protein complex, and they are in between two cells. And as I said before, here below you will find the integrins. Okay, that we don't talk in cell biology. I left it for cancer biology because there is too much to talk in cell biology. So they are so many functions. They are involved in regulating cell shift, mobility, and progression to the cell cycle. So cancer, all the signals can come through all these junctions, and they are very important. And if the cell junctions are not working properly, they could be an important step for cancer development. Uh, so for example, a cell to escape to go to a different region, it needs to take, imagine, it's like you go and cut all these, yeah, you need to cut all these junctions and disassemble them to be able to escape and go to another tissue. So studying these structures is very important to understand how this happens, yeah, how cancer can travel from one region into another one. So now, don't going to go into a lot of detail, but I'm going to tell you that the adhesion junctions, again, similar to integrins, anchor to the cytoskeleton. So in that way, they can, if there is a change in the adhesion of the cells, they can tell to the nucleus, there is something happened here. You need to express different genes. Yeah. So this is why here you see that they are attached to the cytoskeleton to actin. Okay, and here I'm just going to show you a quick video about cell adhesion. Yeah.
After its translation from mRNA, the classic cadherin protein is transported to the cell membrane. The cadherin establishes itself as a transmembrane protein with distinct cytoplasmic and extracellular domains. As seen in this conceptual model, the extracellular segment, which is responsible for the cadherin's ability to link adjacent cells presenting cadherins of compatible type, includes 5 characteristic domains. These cadherin domains interact with domains on neighboring cells in a calcium-dependent manner. In the presence of calcium ions, individual cadherins pair up to form dimers on one side of the intercellular junction. These cis dimers then bind with other dimers on the opposite or trans side. When calcium is removed from the environment, the cadherin molecules fail to bind, and cell-to-cell adhesion is disrupted. The cytoplasmic tail remains within the cell and forms a complex with a trio of catenin molecules, P120, alpha, and beta-catenin. This molecular complex links the transmembrane cadherin protein to the actin cytoskeleton. The cadherin-catenin complex is also important in cell-to-cell adhesion, as cells lacking catenins or expressing cadherins without catenin-binding domains fail to adhere as normal.
Okay, so I have give you so much information. Just this is the last component. So we talk that in the video they talk that there is the one of these molecules involved with adhesion are these E-coherents, yeah, and they are proteins that are outside of the cells and they attach to each other. They talk also that similar to integrins, sorry, they are not only outside, but they have a membrane component, transmembrane component, and they also have a component that is inside of the cell. Yeah, so they go from outside to the membrane to outside. If you see, there's this blue section, and in addition to that, they interact with other molecules that it will help them to attach to the actin. Yeah, cytoskeleton. So here you have this beta-catenin that we talked before. Okay, so good. I know it is a lot of information. I just summarize. I think in this lecture, like I don't know, uh, 20 lectures of cell biology, genetics, and developmental biology. You don't need to memorize everything. I just want to give you tools and that you remember. Some of you remember some of those topics. Some of you will see it later, and you can watch it multiple times. I just want to get you ready for the next topics that they will be here. There will be not a lot of details. I just want that you are aware that all these things exist. Of course, this is more complicated. So you have homophilic cell adhesion molecules, so both are similar. Some of them it could be heterophilic, that they could be that these molecules are different, and depending on which proteins you will have different type of cell adhesion. And this is important to understand development, and this is also important to understand cancer. Uh, this you will hear about this word CAMs, that are cell adhesion molecules like coherence and actin. And again, same thing that I told you, outside of the cell, transmembrane component, and here the intracellular domain. So, and here they fuse with many molecules that they will be finally attached to the cytoskeleton. Okay, so it's the same ideas that we have talked with the integrins. Okay, so I give you review of the main pathways involving cancer, in pancreatic cancer, and I hope that you are not too overwhelmed. Remember, you don't have to memorize every single molecule. I want that you, now that you hear cell adhesion, oh yes, this is very important, and that you hear about something, that you hear about this pathway, you know more or less what it is. Okay, so I hope that you enjoyed this, and I will see you in the next class.